Each time a cell divides, it must first make copies of its DNA to pass on to daughter cells. This process is called DNA replication. In this section, you will learn about the structure of a DNA molecule and how these large macromolecules are packaged to fit within the confines of a cell. You will also learn about the process of DNA replication, in which identical daughter strands are made to promote genetic continuity. All organisms contain genetic material in the form of DNA. It is a defining characteristic that distinguishes living organisms. DNA serves as a template for making proteins and RNA. These instructions are contained in the genetic code. The structure of a DNA molecule enables it to direct the production of proteins throughout the life of a cell. DNA replication is accomplished using several enzymes, each with a specific role. DNA polymerase is the enzyme complex that synthesizes the new daughter strands, shown in blue, during the replication process. Free nucleotides form complementary base pairs with the exposed template strand, and then DNA polymerase forms a bond between them to form a daughter strand. DNA polymerase reads or moves along the template strand in a 5' to 3' direction, and synthesizes the daughter strand in a 3' to 5' direction. Accurate replication of a cell's genetic material is of vital importance to the survival of its daughter cells. After each nucleotide is added to the daughter strand, most DNA polymerases proofread the daughter strand and correct any mistakes that have been made. The process of producing cellular proteins requires a second nucleic acid, RNA. In this section, you will learn about the structure of RNA molecules and how the information contained in DNA is transcribed to form messenger RNA. During transcription, RNA nucleotides line up along the template strand of DNA to form a complementary mRNA molecule. If a template strand of DNA has the base sequence TAGCCA, the complementary mRNA will have the sequence AUCGGU. Remember, RNA nucleotides do not contain thymine, so the nitrogenous base uracil is complementary to adenine. The mRNA transcribed from DNA must become associated with a ribosome, which is the site of protein assembly. In this section, you will learn how the information encoded in mRNA is translated into a polypeptide sequence.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Translation is the synthesis of a polypeptide by a ribosome using the genetic information carried in an mRNA molecule. The ribosome reads the mRNA in groups of 3 nucleotides called codons, beginning with the start codon. Each codon in the coding region of the mRNA specifies a particular amino acid. Of the 64 possible codons, 62 code for one of 20 possible amino acids, and the remaining 2 are stop codons.